Welcome everybody. My name is Leó Asztalos and I am a science student for 3 years. During this time, my field of research included investigating HLA molecules and their role in anti-tumor immunity. Cancer is one of the leading causes of death worldwide. Globally, it is responsible for around 1 out of 6 deaths each year. This is about 10 million deaths. But what exactly is cancer? Our cells have a well-written program, the DNA, that tells them when and in what extent should they divide. If this program goes wrong, some of our cells may begin to divide in an uncontrolled manner and tumors develop. For simplicity, I will use the terms tumor and cancer as synonyms. I am sure that you already knew that cancer is a life-threatening disease. But did you know that tumorous cells occur in our body almost every day? They do, but they just rarely survive. One key element in destroying tumor cells is our immune system. For example, T cells are part of our adaptive immune system. Their task is to destroy dangerous entities like tumorous cells, but they also have to tolerate our normal cells. To do this effectively, they have to differentiate the tumor cells from the rest of our cells. This is possible through immune recognition. The basis of immune recognition in tumors is that the mutation of the DNA results in mutant proteins. These mutant proteins get processed into smaller pieces, and we call these peptides. These peptides can be bound to HLA molecules on the cell surface. T cells can then recognize these peptides bound to HLA molecules, and if it happens so, the T cells will destroy the tumorous cells. This mechanism forms the basis of anti-tumor immunity. We can utilize this mechanism in many ways. It opened the way for many new therapies called immune therapies. One such therapy is our anti-tumor vaccines. In this case, we collect mutant peptides and inject them into the patient. After this, these mutant peptides will boost the patient's immune system and it will be able to destroy the tumor cells more effectively. And another therapy is called checkpoint blockade immunotherapy. Tumor cells develop various tactics to evade the destruction by the immune system. One such tactic is the formation of inhibitory molecules that block the normal function of the T cells. But luckily, with checkpoint blockade immune therapy, we can block these inhibitory molecules, and therefore T cells will regain their normal function and they will effectively destroy the tumor cells. As you can see, in both cases, the immune recognition is very important because T cells can destroy tumorous cells through the recognition of mutant peptides bound to HLA molecules. HLA molecules are outstandingly variable. Actually, they are the most variable region of the human genome. There are many thousand different registered HLA variants, and 2 individuals potentially carry different HLA variants. These molecules differ in their peptide binding ability, which means that the HLA molecules of 2 individuals bind a different number of peptides. And this difference in the number of bound different peptides affects anti-tumor immunity too. So there are many thousand different HLA variants, and there are approximately 25,000 protein coding regions in the genome. In every gene, mutations can result in mutant proteins. And this relationship affects how effective the anti-tumor immunity will be and how effectively an immune therapy will be in the case of some patients, which can decide between life and death. In our laboratory, we use modern computational technologies and combined it with immunology. This discipline is called computational immunology. With the help of computer programs, we analyze big databases that contain enormous amounts of data. And with the help of this approach, we can investigate complex systems and not only solitary genes or molecules. In our latest research, we investigated the effect of different HLA variants on anti-tumor immunity. We categorized the HLA molecules into two groups. In the first group, the HLA molecules were able to bind a big number of different peptides, and therefore we called these generalist HLA variants. In the other group, the HLA molecules were able to bind only a small number of different peptides, and therefore we call these specialist HLA variants because they bind only a small and specific set of peptides. In the case of generalist HLA molecules, we expected that because of the more different bound peptides, it will result in a more effective anti-tumor immunity. In the case of specialist HLA variants, we expected that because of the small number of different bound peptides, there will be a less effective anti-tumor immunity. Surprisingly, we found the opposite of what we expected. We investigated the survival of cancer patients treated with immunotherapies, and we found that patients carrying generalist HLA variants survived worse than the patients who carried specialist HLA variants. On this figure, you can see the survival of the patients, and the red line shows the survival of patients carrying generalist HLA variants, and the blue line shows the survival of patients carrying specialist HLA variants. As you can see, in both cases, patients carrying specialist HLA variants, therefore binding less different peptides, survived better. But how can this be explained? Mutant proteins develop from our normal proteins, and in many cases, there is just a small difference between them. In the case of generalist HLA molecules, because they bind many different peptides, they can bind the mutant and the normal proteins too. Therefore, our immune system may not recognize the slight changes between the normal and the mutant proteins. Because of this, this will result in a less effective anti-tumor immunity. In the case of specialist HLA molecules, because they bind only a small number of different peptides, if they bind only the mutant protein, then the immune system will recognize these and it will effectively destroy the tumor cells. This will result in a more effective and anti-tumor immunity. So as you could see, research is very adventurous because you never know where you will end up. It is also a lot of work, but scientific research is always a teamwork, and here you can see the core members of our laboratory. It is very, for me, it is very exciting that we are researching something unknown and that it may change the lives of many people. If you are interested in our work, visit our website where you can find out more about our research group. And if you have any questions, feel free to contact me on this email. Thank you for your attention.